Students, let's get started with the section 2 of Harpers Illustrated Biochemistry, which is enzymes, kinetics, mechanism, regulation, and role of transition metals. It covers the chapter 6 to 10th of your textbook. So, starting off with chapter 6, proteins, myoglobin, and hemoglobin. The objectives of this chapter are so that you can be able to describe the most important structural similarities and differences between myoglobin and hemoglobin, sketch binding curves for the oxygenation of myoglobin and hemoglobin, explain why hemoglobin's sigmoidal O2 binding curve renders it a better vehicle for transporting oxygen than myoglobin's hyperbolic binding curve, explain how the presence of distal histidine affects the ability of hemoglobin to bind carbon monoxide CO, define P50 and indicates its significance in oxygen transport and delivery, describe the structural and conformational changes in hemoglobin that accompany its oxygenation and subsequent deoxygenation, what role does the proximal histidine play in this transition, explain the role of 2,3-BPG in oxygen binding and delivery, explain how Bohr effect enhances the ability of red blood cells to absorb CO2 from peripheral tissues and promotes the release of transported CO2 in the O2 rich environment of the lungs. Describe the structural consequences to hemoglobin S, HPS of lowering PO2 and identify the metabolic defects that occur as a consequence of alpha and beta thalassemias. So, starting with biomedical importance. The efficient delivery of oxygen from lungs to the peripheral tissues and the maintenance of the tissues reserves to protect against anoxic episodes are essential to health. In mammals, these functions are performed by homologous heme proteins, hemoglobin and myoglobin, respectively. Myoglobin, a monomeric protein of red muscle, binds oxygen tightly as a reserve against oxygen deprivation. The multiple subunits of hemoglobin, a tetrameric protein of erythrocytes, interact in a cooperative fashion that enables this transporter to offload a high proportion of bound O2 in peripheral tissues while simultaneously retaining the capacity to bind it efficiently in the lungs. The offloading of oxygen is enhanced by the binding of 2,3-BPG, which stabilizes the quaternary structure of deoxyhemoglobin. Cyanide and carbon monoxide CO kill because they disrupt physiologic function of heme protein, cytochrome oxidase, and hemoglobin respectively. In addition to delivering O2, hemoglobin plays a critical role in the transport of waste product of respiration, CO2, to the lungs for disposal. Proton binding to hemoglobin enhances the conversion of CO2, a major product of respiration, into water-soluble carbonic acid and its conjugate base, bicarbonate, by the enzyme carbonic anhydrase. In the lungs, O2-induced proton release from hemoglobin reverses this process to facilitate disposal as gaseous CO2. Additional CO2 is carried in the form of covalently bound carbamates. Hemoglobin and myoglobin illustrate both protein structure function relationships and the molecular basis of genetic disorders such as sickle cell disease and the thalassemias. So, starting with heme and ferrous iron confer the ability to store and transport oxygen. Myoglobin and hemoglobin contain heme, an iron-containing tetracyclic the cyclic tetrapyrrole consisting of 4 molecules of pyrrole linked by methine bridges. This planar network of conjugated double bonds absorbs visible light and colors heme deep red. The substituents at the beta positions of heme are methyl M, vinyl V, and propionate PR groups arranged in the order of M, V, M, V, M, PR, PM, M, like we can see in 6.1. We can see that the pyrrole rings and methylene bridges carbons are coplanar and the iron atom Fe2 plus resides in almost the same plane. The fifth and sixth coordination position of Fe2 plus are directly perpendicular to and directly above and below the plane of the heme ring. Observe the nature of methyl blue, vinyl green, and propionate orange substituent groups on the beta carbons of the pyrrole rings, the central iron atom red, and the location of the polar side of heme ring at about 7 o'clock that faces the surface of the myoglobin molecule. Okay. An atom of ferrous iron Fe2 plus resides at the center of the planar tetrapyrrole. Oxidation of the Fe2 plus of myoglobin or hemoglobin to Fe3 plus destroys their biological activity. Other proteins with metal containing tetrapyrrole prosthetic groups include cytochromes, Fe and Cu, and chlorophyll Mg. Myoglobin is rich in alpha helix. Oxygen stored in red muscle myoglobin is released during O2 deprivation, example severe exercise, for use in muscle mitochondria for aerobic synthesis of ATP. A 153 amino acyl residue polypeptide molecular weight 17,000, the compactly folded myoglobin molecule measures 4.5 into 3.5 into 2.5 nm, like we can see in the diagram. The three-dimensional structure of myoglobin shows a ribbon diagram tracing the polypeptide backbone of myoglobin. The color of the polypeptide chain is graded along the visible spectrum from blue N terminal to tan C terminal. The heme prosthetic group is red. The alpha helical regions are designated A through H. The distal E7 and proximal Fa histidine residues are highlighted in blue and orange, respectively. Note how polar propionate and propionate substituents PR project out of the heme toward the solvent. Adapted. Okay. So, coming back, an unusually high proportion, about 75% of the residues are present in 8 right handed 7 to 20 residue alpha helices. Starting at the amino terminal, these are termed as helices A through H. Typical of globular proteins, the surface of myoglobin is rich in amino acids bearing polar and potentially charged side chains, while the two exceptions, the interior contains residues that possess non-polar R groups, example leucine, valine, phenylalanine, and methionine. The exceptions are the 7th and 8th residues in helices E and F, his E7 and his F8, which lie close to the heme iron, the site of O2 binding. Histidine F8 and E7 perform unique roles in oxygen binding. The heme of myoglobin lies in a crevice between helices E and F, oriented with its polar propionate groups facing the surface of the globin. The remainder resides in the nonpolar interior. It is held in place mainly by the hydrophobic interactions. The fifth coordination position of iron is occupied by nitrogen from the imidazole ring of the proximal histidine, his F8. The distal histidine, his E7, lies on the side of the heme ring opposite to his F8. In figure 6.3, we can see the angles for bonding of oxygen and carbon monoxide to the heme iron of myoglobin. The distal E7 histidine hinders bonding of CO at the preferred 90-degree angle to the plane of heme ring. The iron moves toward the plane of the heme when oxygen is bound. The iron of unoxygenated myoglobin lies 0.3 nm, zero point 0.3 angstrom outside the plane of the heme ring towards the proximal histidine, his F8. Consequently, the heme puckers slightly. When O2 occupies the sixth coordination position, the iron moves to within 0.01 nm, zero point 0.1 angstrom to the plane of the heme ring. Oxygenation of myoglobin thus is accompanied by motion of the iron of his F8 and residues linked to his F8. Apomyoglobin provides a hindered environment for the heme iron. Minute quantities of CO arise from a variety of biologic sources, including the catabolism of red blood cells within the human body. CO is also present in the atmosphere, mainly due to the incomplete combustion of fossil fuels. CO binds to the iron in a free heme group 25,000 times more strongly than globin than oxygen. However, the heme groups in the apoproteins of myoglobin and hemoglobin reside in a hindered environment for their gaseous ligands. Figure 6.3. When CO binds to free heme, all three atoms Fe, C, and O lie perpendicular to the plane of the heme. This geometry maximizes the overlap between the lone pairs of electrons on the sp hybridized carbon of the CO molecule and the Fe2 plus iron. Figure 6.4 we can see. Orientation of the lone pairs of the electrons relative to the O double bond O and C triple bond O bonds of oxygen and carbon monoxide. In molecular oxygen, formation of double bond between the two oxygen atoms is facilitated by the adoption of sp2 hybridization state by the valence electron of each oxygen atom. As a consequence, the two atoms of the oxygen molecule and each lone pair of electrons are coplanar and separated by angle of 120 degree. By contrast, the two atoms of carbon monoxide are joined by a triple bond which requires the carbon and oxygen atoms adopt a sp hybridization state. In this state, the lone pairs of electrons and triple bonds are arranged in a linear fashion where they are separated by an angle of 180 degree right. By contrast, O2 is sp2 hybridized. Consequently, the electrons that bind to the iron lie at an angle of roughly 120 degree to the axis of the O double bond O. In myoglobin and hemoglobin, the distal histidine sterically precludes or hinders the preferred high affinity orientation of CO, while still permitting O2 to attain its most favorable orientation. Binding at less favored angle reduces the strength of heme CO bond to about 200 times that of the heme O2 bond. Therefore, O2, which is typically present at greater excess over CO, normally dominates. Nevertheless, about 1% of myoglobin typically is present combined with CO. The oxygen dissociation curves for myoglobin and hemoglobin suit their physiological roles. The hyperbolic relationship between the concentration or partial pressure of O2, PO2, and the quantity of O2 bound expressed as O2 saturation isotherm reveals why myoglobin is more suitable for O2 storage than for O2 transport. Myoglobin loads O2 readily at the PO2 of the lung capillary bed 100 mmHg. However, since myoglobin releases only a small fraction of this bound O2 at the PO2 values typically encountered in active muscle 200 mmHg or other tissues 40 mmHg, it represents an ineffective vehicle for delivery of O2. However, when strenuous exercise lowers the PO2 of muscle tissue to about 5 mmHg, dissociation of O2 from myoglobin permits the mitochondrial synthesis of ATP and hence muscle activity to continue. Okay. In figure 6.5, we can see that oxygen binding curves of both myoglobin and hemoglobin are given. The arterial oxygen tension is about 100 mmHg, mixed venous oxygen tension is about 40 mmHg, capillary active muscle oxygen tension is about 20 mmHg, and the minimum oxygen tension required for cytochrome oxidase is about 5 mmHg. Association of the chains into tetrameric structure hemoglobin results in much greater oxygen delivery than would be possible with single chains. By contrast, hemoglobin behaves as if it were two proteins. At the high PO2 of lungs, 100 mmHg and above, it displays a high affinity for oxygen that enables it to bind oxygen to nearly every available heme ion. This form of protein is commonly referred to as R for relaxed state hemoglobin. At lower PO2 values encountered in peripheral tissues, 40 mmHg and below, hemoglobin exhibits a much lower apparent affinity for oxygen. Transition of hemoglobin to this low affinity TOUT or T state enables it to release a large proportion of oxygen previously picked up in the lungs. This dynamic interchange between the high and low affinity R and T states serves as the foundation for hemoglobin's sigmoidal O2 binding curve. The allosteric properties of hemoglobins result from their quaternary structures. The quaternary structure of hemoglobin confers striking additional properties absent from monomeric myoglobin, which adapts this tetrameric protein to its unique biological roles in the reciprocal transport of O2 and CO2 between the lungs and peripheral tissues. Hemoglobin is tetrameric. Hemoglobins are tetramers composed of pairs of two similar but distinct polypeptide subunits, as we can see in this figure. Hemoglobin shown is the three-dimensional structure of deoxyhemoglobin with a molecule of 2,3-BPG dark blue bound. The two subunits are colored in the darker shades of green and blue, the two subunits with the lighter shades of green and blue, and the heme prosthetic group in red adapt. Okay. So, Greek letters are used to designate each subunit type. The subunit composition of principal hemoglobin are alpha 2, beta 2, HbA normal adult hemoglobin, alpha 2, gamma 2 in HbF fetal hemoglobin, alpha 2, beta 2 S HbS sickle cell hemoglobin, and alpha 2, delta 2 HbA2 minor adult hemoglobin. The primary structures of beta, gamma, and delta chains of hemoglobin are highly conserved. Myoglobin and the beta subunits of hemoglobin share almost identical secondary and tertiary structures. Despite the differences in the kind and number of amino acids present, myoglobin and the beta polypeptide of hemoglobin A share almost identical secondary and tertiary structures. Similarities include the location of heme and the helical regions and the presence of amino acids with similar properties at comparable locations. Although it possesses 7 rather than 8 helical regions, the alpha polypeptide of hemoglobin also closely resembles myoglobin. Oxygenation of hemoglobin triggers conformational changes in the apoprotein. Hemoglobins can bind up to 4 molecules of O2 per tetramer, 1 per heme. However, once the first molecule of O2 becomes bound, the affinity of the other three subunits increases. Termed cooperative binding, this behavior permits the hemoglobin to maximize both quantity of O2 loaded and at the PO2 of the lungs and the quantity of O2 released at the PO2 of peripheral tissues. P50 expresses the relative affinities of different hemoglobins for oxygen. The quantity P50, a measure of O2 concentration, is the partial pressure of O2 at which given hemoglobin reaches half saturation. Depending on the organism, P50 can vary widely. But in all its instances, it exceeds the normal PO2 of peripheral tissues. For example, the values of P50 for HbA and HbF are 26 and 20 mmHg respectively. In the placenta, this difference enables HbF to extract oxygen from HbA in the mother's blood. However, HbF is suboptimal postpartum since its higher affinity for O2 limits the quantity of O2 delivered to the tissues. The subunit composition of hemoglobin tetramers undergoes complex changes during development. The human fetus initially synthesizes epsilon 2E2 tetramer. By the end of first trimester, epsilon and E subunits have been replaced by alpha and delta subunits, forming HbF alpha 2 gamma 2. The hemoglobin of late fetal life. Now, while synthesis of beta subunits begins in the third trimester, the replacement of gamma subunits by beta subunits to yield adult HbA alpha 2 beta 2 does not reach completion under several weeks postpartum. See figure 6.7. Like developmental pattern of quaternary structure of fetal and newborn hemoglobin can be seen. Like the fetal gamma chain is depleting and the beta chain is arising. In figure 6.8, we can see on oxygenation hemoglobin, iron atom moves to the plane of heme. Histidine F8 and its associated amino acid residues are pulled along with the iron atom. Oxygenation of hemoglobin is accompanied by large conformational changes. The binding of first molecule of O2 to deoxy-Hp shifts the heme iron towards the plane of the heme ring. This motion is transmitted through the proximal F8 histidine and the residues attached thereto onto the entire tetramer, triggering the rupture of salt bridges formed by the carboxyl terminals residues of all four subunits. As a result, one pair of alpha-beta subunits rotates 15 degrees with respect to the other, compacting the tetramer. In figure 6.9, we can see during transition of the T form to the R form of hemoglobin, alpha to beta 2 pair of subunits rotates to 15 degree relative to the pair of alpha 1 beta 1 subunits. Okay, the axis of rotation is eccentric and the alpha to beta 2 pair also shifts toward the axis somewhat. In the representation, the tan alpha 1 beta 1 pair is shown fixed, while the green alpha to beta 2 pair of subunits both shifts and rotates. Now, this triggers the other. Profound changes in secondary, tertiary, and quaternary structure that accompany the transition of hemoglobin from the low affinity T state to high affinity R state. These changes significantly increase the affinity of the remaining unoxygenated hemes for O2. Subsequent binding events require the rupture of fewer salt bridges, like in figure 610. Can see transition from the T structure to the R structure. In this model, salt bridges linking the subunits of in the tree structure break progressively as oxygen is added, and even those salt bridges that have not yet ruptured are progressively weakened wavy red lines. The transition from T to R does not take place for a fixed number of oxygen molecules have been found out, but becomes more probable as each oxygen binds. The transition between the two structures is influenced by protons, carbon dioxide, chloride, and 2,3-BPG. The higher their concentration, the more oxygen must be bound to trigger the transition. Fully oxygenated molecules in the T structure and fully deoxygenated molecules in the R structure are not shown because they are unstable. The cooperativity of hemoglobin constitutes a form of allosteric behavior, since binding of O2 to one heme affects the binding affinity of the others. Hemoglobin assists in the transport of CO2 to the lungs. For every molecule of O2 consumed during the course of respiration, one molecule of CO2 is produced. Consequently, red blood cells must be able to efficiently absorb CO2 from respirating tissues and subsequently release it for disposal in the lungs. Two key contributors to this process are the enzymatic carbonic anhydrase, which catalyzes the dehydration of CO2 to water-soluble carbonic acid H2CO3, and the ability of hemoglobin to cooperatively transition between T and R states. Carbonic anhydrase converts CO2 to water-soluble carbonic acid and bicarbonate. As is the case for O2, the limited water solubility of CO2 at neutral pH renders simply dissolving this gas in the bloodstream grossly insufficient to meet the body's transport needs. The enzyme carbonic anhydrase, present in large quantities in red blood cells, catalyzes the dehydration of CO2 molecules to form water-soluble carbonic acid H2CO3. H2CO3 is a weak acid that in turn can dissociate to form bicarbonate ion HCO3- and a proton. Since the pKa1 of H2CO3 6.35 falls below the physiologic pH in the red blood cells, the majority of absorbed CO2 is carried as water-soluble bicarbonate. Binding of proteins to T-state hemoglobin increases CO2 uptake from respiring tissues. When hemoglobin transitions from the R to the T state, conformational changes take place in each of the two beta chains that lead to the formation of salt bridges between the side chains of ASP94 and His146. Formation of each salt bridge requires T-state hemoglobin to bind a proton from the surrounding environment. As R-state hemoglobin gives up its bound O2 to respiring tissues and subsequently transitions to the T state, the absorption of these two protons both buffers the pH of the acidifying red blood cell and shifts or pulls the equilibrium between H2CO3 and HCO3- in favor of bicarbonate, further increasing the quantity of carbon dioxide absorbed by the red blood cells from peripheral tissues. T-state hemoglobin binds 2 protons per tetramer. In actively respiring tissues, the proton concentration inside the red blood cells will increase as H2CO3 accumulates. The greater availability of H plus in turn favors the formation of T-state hemoglobin, thereby enhancing the release of O2. Proton binding T-state hemoglobin enables the high levels of CO2 in actively respiring tissues to drive the release of O2 from hemoglobin while concomitantly enhancing the quantity of CO2 absorbed by promoting the conversion of carbonic acid to bicarbonate. As a result, transition to the T state helps buffer or moderate the CO2 mediated decrease in the pH of the red blood cells in venous blood. Proton release from R-state hemoglobin enhances CO2 release in the lungs. On reaching the lungs, the dramatic increase in the partial pressure of oxygen drives the binding of O2 to deoxyhemoglobin. O2 binding in turn triggers the transition of hemoglobin from the T to R state. Resulting conformational changes causes the rupture of salt bridges between the two beta chains and the release of two protons formerly chelated between ASP94 and His146. Once freed, the protons combine with bicarbonate to increase the concentration of carbonic acid, which in turn favors carbonic anhydrase catalyzed dehydration of H2CO3 to form CO2, which can then be disposed by exhalation. We can see in figure 6.11. We'll discuss this. This proton-mediated coupling of the transition of hemoglobin between T and R states with the equilibria between CO2, H2CO3 and HCO3- is known as the Bohr effect. In figure we can see that carbon dioxide generated in the peripheral tissues combines with water to form carbonic acid, which dissociates into protons and bicarbonate ions. Deoxyhemoglobin acts as buffer by binding protons and delivering them to the lungs. In the lungs, the uptake of oxygen by hemoglobin releases protons that combine with the bicarbonate ion, forming carbonic acid, which when dehydrated by carbonic anhydrase becomes carbon dioxide, which then is exhaled. Additional CO2 is transported as carbamates of hemoglobin. About 15% of CO2 in venous blood is carried by hemoglobin as carbamates formed with the amino terminal nitrogens of its polypeptide chains. CO2 plus HbNH3 plus yields 2 protons and the compound. Carbamate formation changes the charge on amino terminals from positive to negative, favoring salt bridges formation between alpha and beta chains. As part of Bohr effect, proton binding T state hemoglobin favors carbamate formation, while proton release on transition to R state favors carbamate breakdown and release of CO2. 2,3-BPG stabilizes the T structure of hemoglobin. The transition of hemoglobin to the T states open a central cavity at the interface of the four subunits capable of binding one molecule of 2,3-BPG. Here we can see in figure 6.6, okay, or above. Binding of 2,3-BPG thus favors the shift from the R state to T state and the further release of bound O2. As conversion back to the R state requires the rupture of the additional salt bridges formed between 2,3-BPG and lyse EF6 is H21 and the terminal groups of val Na1 from both beta chains. Figure 6.12. Mode of binding 2,3-BPG to human deoxyhemoglobin. BPG interacts with the three positively charged groups of each beta chain. Okay. 2,3-BPG is synthesized from the glycolytic intermediate 1,3-BPG, a reaction catalyzed by the bifunctional enzyme 2,3-bisphosphoglycerate synthase 2-phosphatase BPGM. BPG is hydrolyzed to 3-phosphoglycerate by the 2-phosphatase activity of BPGM and to 2-phosphoglycerate by second enzyme multiple inositol polyphosphate phosphatase MIPP. The activities of these enzymes and hence the level of BPG in erythrocytes are sensitive to pH. The CO2 induced acidification of the red blood cells triggers production of 2,3-BPG, which in turn reinforces the impact of carbonic acid derived protons in shifting the RT equilibrium in the favor of T state, increasing the quantity of O2 released in the peripheral tissues. In the fetal hemoglobin, residue H21 of gamma subunit is SER rather than his. Since SER cannot form a salt bridge, PPG binds more weakly to the HbF than to HbA. The lower stabilization afforded to the T state by BPG helps account for HbF having a high affinity for O2 than HbA. Adaptation to high altitude. Physiologic changes that accompany prolonged exposure to high altitude increases in the number of erythrocytes, the concentration of hemoglobin within them, and the synthesis of 2,3-BPG. Elevated 2,3-BPG lowers the affinity of HbA for O2, increases P50, which enhances the release of O2 at peripheral tissues. Numerous mutations affecting human hemoglobins have been identified. Mutation in the genes that encode the alpha or beta subunits of hemoglobin potentially can affect its biologic functions. However, of more than 1,100 known genetic mutations affecting human hemoglobins, most are both rare and benign, presenting no clinical abnormalities. When a mutation does compromise biological function, the condition is termed hemoglobinopathy. It is estimated that more than 7% of globe's population are carrier for hemoglobin disorders. The URL provides information about the links for normal and mutant hemoglobin. Selected examples are as follows. Methemoglobin and hemoglobin M. In methemoglobinemia, the heme iron is ferric rather than ferrous, rendering the methemoglobin unable to bind or transport O2. Commonly, the Fe3 plus of methemoglobin is assigned to return to Fe2 plus state through the action of the enzyme methemoglobin reductase. Methemoglobin levels can rise to pathophysiologically significant levels from a number of causes. Oxidation of Fe2 plus to Fe3 plus as a side effect of agents such as sulfonamides. Reactions in the activity of methemoglobin reductase. Reduction in the activity of methemoglobin reductase or inheritance of a gene for a mutationally altered form of hemoglobin called HbM. In hemoglobin M, histidine F8, his F8, has been replaced by tyrosine. The iron of HbM forms a tight ionic complex with the phenolate anion of tyrosine that stabilizes the Fe3 plus form. In alpha chain hemoglobin M variants, the RT equilibrium favors the state. Oxygen affinity is reduced and the Bohr effect is absent. Beta chain hemoglobin M variants exhibit RT switching and the Bohr effect is therefore present. Mutations that favor R state hemoglobin Chesapeake increase O2 affinity. These hemoglobins therefore fail to deliver adequate O2 to peripheral tissues. The resulting tissue hypoxia leads to polycythemia and increased concentration of erythrocytes. Hemoglobin S. In HbS, non-polar amino acid valine has been replaced and has replaced the polar surface residue glutamine 6 of the beta subunit. resulting a hydrophobic sticky patch on the surface of beta subunit of both oxy-HBs and deoxy-HBs. Both HBA and HBs contain complementary sticky patch on their surfaces that is exposed only in the deoxygenated T state. Thus, at low PO2, deoxy-HBs can polymerize to form long twisted helical fibers. Binding of deoxy-HBa terminates fiber polymerization since HBa lacks the second sticky patch necessary to bind another HB molecule. Like in figure 6.13 we can see. Uh, uh, these insoluble fibers distort the erythrocyte into a characteristic sickle shape, rendering it vulnerable to lysis by the intrinsics of the splenic sinusoids. They also cause multiple secondary clinical effects. A low PO2, such as that in high altitudes, exacerbates the tendency to polymerize. The term sickle cell trait and sickle cell disease refer to the persons whom gene for either one or both beta subunits are mutated, respectively. Emerging treatment for sickle cell disease include HbF expression to inhibit the polymerization of HbS, stem cell transplantation, and in the future, gene therapy. Biomedical implications. Myoglobinuria. Following massive crush injury to skeletal muscle followed by renal damage, released myoglobin may appear in the urine. A myoglobin can be detected in the plasma following a myocardial infarction, but assay of serum troponin, lactate dehydrogenase enzyme, or creatine kinase provides a more sensitive index of myocardial injury. Anemias. Anemia's reduction in the number of red blood cells or concentration of hemoglobin in the blood can reflect impaired synthesis of hemoglobin, example iron deficiency, or impaired production of erythrocytes, example folic acid or vitamin B12 deficiency. Diagnosis of anemias begins with spectroscopic measurement of blood hemoglobin levels. If we look at this, we can see polymerization of deoxyhemoglobin S. The dissociation of oxygen from hemoglobin S unmasks a sticky patch red triangle on the surface of its beta subunits green that can adhere to complementary site on the beta subunits of the other molecules, deoxy-HBs. Polymerization to a fibrous polymer is interrupted by deoxy-HBa, whose beta subunits in lavender color lack the sticky patch required for binding additional HBs subunits. Coming on to thalassemias. The genetic defect known as thalassemia results from the partial or total absence of one or more alpha-beta chains of hemoglobin. Over 750 different mutations have been identified, but only 3 are common. Either the alpha chain, alpha thalassemia, or beta chain, beta thalassemia can be affected. The superscript indicates whether a subunit is completely absent, alpha 0 or beta 0, or whether synthesis is reduced, alpha negative or beta negative. Apart from marrow transplantation, treatment is symptomatic. Certain mutant hemoglobins are common in many populations, and patient may inherit more than one type. Hemoglobin disorders thus present a complex pattern of clinical phenotypes. The use of DNA probes for diagnosis is considered in later chapters. Glycated hemoglobin HbA1c. Blood glucose that enters the erythrocyte can form a covalent adduct with the epsilon amino groups of lysine residues and the alpha amino group of N-terminal valine of hemoglobin beta chains, a process referred to as glycation. Unlike glycation is not enzyme catalyzed. The fraction of hemoglobin glycated, normally about 5%, is proportionate to blood glucose concentration. Since the lifespan of an erythrocyte is typically 120 days, the level of glycated hemoglobin HbA1c reflects the mean blood glucose concentration over the preceding 8 to 12 weeks. Measurement of HbA1c therefore provides valuable information for management of diabetes mellitus. So coming to the summary of this chapter, myoglobin is monomeric. Hemoglobin is a tetramer of two subunit types, alpha 2 beta 2 in HbA. Myoglobin and the subunits of hemoglobin are homologues of one another that, despite differences in primary structure, exhibit nearly identical secondary and tertiary structures. Heme is an essentially planar, slightly puckered cyclic tetrapyrrole whose 4 nitrogen atoms bind a central Fe2+ that is also linked to histidine F8 and in oxymethemoglobin and oxyhemoglobin to O2. The O2 binding curve for myoglobin is hyperbolic, but for hemoglobin it is sigmoidal as a consequence of cooperative interactions in the tetramer. Cooperativity arises from the ability of hemoglobin to exist in two different conformational states, the relaxed or R state, in which all the 4 subunits exhibit a high affinity for oxygen, and a taut or T state where all the 4 subunits display a low affinity for oxygen. The high levels of O2 in the lungs drive the RT equilibrium in the favor of R state, while acidification of the red blood cells generated from the catalytic hydration of CO2 in the peripheral tissues favors the T state. Cooperativity thus maximizes the ability of hemoglobin both to load O2 and PO2 of the lungs and to deliver O2 and PO2 of the tissues at the PO2 of the tissues. Relative affinities of different hemoglobins of oxygen are expressed as P50, the PO2 at which they become half saturated with O2. Hemoglobin isoforms saturate at the partial pressure of the respectively respiratory organ, for example, the lung or placenta. On oxygenation of hemoglobin, the iron and histidine F8 move toward the heme ring. The resulting conformational changes in the hemoglobin tetramer induce the rupture of several salt bonds, the release of bound protons, and loosening of the quaternary structure that increases the affinity for the other three subunits of O2. Binding of 2,3-BPG in the central cavity of T state hemoglobin favors O2 release in actively respiring tissues. Transition to the R state results in the closure of this cavity and extrusion of 2,3-BPG. Hemoglobin assists the CO2 transport from peripheral tissues to the lungs via the formation of carbamates and the Bohr effect, a consequence of binding of protons to T state but not to R state hemoglobin. Proton binding enhances the conversion of CO2 to water soluble carbonic acid and bicarbonate. In the lungs, the release of protons from oxygenated R state hemoglobin favors the conversion of bicarbonate and carbonic acid to CO2, which is exhaled. In sickle cell, hemoglobin HbS well replaces glue-6 of the beta subunit of HbA, creating a sticky patch that has a complement on deoxy-HB but not on oxy-HB. Deoxy-HbS polymerizes at low O2 concentrations, forming fibers that distort erythrocytes into sickle shape. Alpha and beta thalassemias are anemias that result from reduced production of alpha and beta subunits of HbA, respectively. So this completes our chapter 6. Okay, like and subscribe. Thanks for watching.